Hello. All right. So welcome back to the coffee break with TRANSFAC. Today is a very important topic. Basically, let me share the screen. All right. So your first TRANSFAC analysis. But first question is why? Why do you need to actually analyze using TRANSFAC? And I have a good example here. With a snake. It's a published quite old paper, but very interesting in Cell, where it's it underlines the importance of transcription factor binding sites. Of course, everybody knows transcription factors are important. If you have in the genome you study, you get a mutation in a transcription factor, which regulates hundreds of thousands of genes. Of course, you get a very severe change, and maybe it leads to a disease, or even this organism cannot live at all. But what happens is when a single transcription factor binding site is mutated, and it's like a little area in genome, one nucleotide is changed, so it's like in billions of nucleotides. Who cares? But no, and sometimes they're very important. For instance, in this enhancer, there's one place with this little transcription factor binding site for this factor ETS1, which actually control whether you will have your lags or not. Because what these people did, they actually compared this enhancer region between mouse and snake genome and found this difference. And when they cut this enhancer from mouse, they actually see mouse basically with very truncated legs, and even in the very beginning of the development. And other way around, if they actually put it back, so this enhancer again, so then they recover the development of legs. So that shows you how important is this to predict transcription factor binding sites. But before we going into more details about the theory of how we do that and why, and what is the algorithm, and how to apply it, let me first actually just go to the tool we have, to TRANSFAC, and to start from scratch. Basically you have your tool, and let's do the first TRANSFAC analysis. Okay, so for that I will go to the website. Here is a website, and to log into website, you to our tool, you have to click here. Here is a login, and you have your account definitely if you are here. If not yet, so you can register here to get your account. When at least you get account to the platform we are now going to present. And also you can apply for TRANSFAC if you don't have it yet. So, but first you have to have this user account. So you sign in, and then you have the list of products. So you have this TRANSFAC database, and of course, you would go to TRANSFAC and do your search to about the genes, how they are regulated, these transcription factors, what is known about them. Yeah, so for instance, you're maybe interested in the transcription factor, which is encoding a component of a transcription factor called NF-κB. It's a very important transcription factor. It's involved in regulation of many inflammatory processes, and not only many processes in the cell. It regulates, and then you can read about this factor. For instance, you can understand where it is expressed, what are the other features of that factor. So you see, for instance, which are known mutations, what is expression level in different tissues, and if this factor is expressed from a particular gene, and you can also learn how this gene is regulated. So basically what you see here are those transcription factor binding sites for other transcription factors which regulate expression of this one transcription factor. So they are kind of standing upstream from this factor, all right? So, and these binding sites are actually known. They are experimentally proven and they are in TRANSFAC database. So you can read about them, how they were proved, whether it was like a ChIP-seq experiment, not massive, but really particular to a particular place in genome, or maybe more intense study of those sites were done. So all this information is stored in this database. But then you can go further and read about this transcription factor, and particularly a very important part which is interesting for many people who are using TRANSFAC are those, we call them motifs. So actually those motifs in genome, which are places to bind, to play, well, where transcription factor NF-kappa B is binding. And you can see here in TRANSFAC, for instance, for this REL A component transcription factor, for NF-kappa B transcription factors, we have 11 matrices, 11 motifs, and each motif is described like a matrix. So, and we'll talk about this today, like in more theoretical way. So, but for now, you just need to know that it's not just a consensus which we store here in TRANSFAC. It's actually information about frequency of each nucleotide happen in each position of that sequence, which is called the binding site for NF-kappa B. And those matrices are built from by us, just by collecting binding sites from various experiments, or maybe they're like in this case, they are collected from one Celix experiment. So these binding transcription factor matrices, they are built in TRANSFAC, and currently we have more than 10,000 matrices in TRANSFAC, which covers many organisms and many families of factors. Actually, for human, we can say that we cover almost cover actually more than 97% of transcription factors of human. So each of that factor has at least one matrix in TRANSFAC. There are a few which are not yet have, but there are very strange factors. Nobody knows about them, anything. But for majority of them, we can associate a particular transcription factor matrix. But okay, so we have all this collection of matrices, but now we want to predict binding sites for these sites, for these factors in new regions of genome. So we know some regions are binding by these factors. This we know from various experiments, but as you can understand, and we make this assumption, and we actually make these predictions that almost 99% of sites which are actually actual binding sites in genome, and in some conditions transcription factors of various nature can bind to these places, but they are not yet known. So neither in TRANSFAC nor in any ChIP-seq experiments. So majority of sites are still unknown. That's a very important fact. So even if you take all ChIP-seq experiments which are done so far, still 99% are still missing. Yeah. So which means you have to predict transcription factor binding sites, and why you should do it. As I mentioned in the beginning, it's very important. Even one binding site which you not predict, which you may miss for some reason, may be very crucial, like you have a LEX or you don't have LEX. All right. So, but how to predict transcription factor binding sites? And we have the tools, various tools to do that. So we are working on that for many years. For TRANSFAC itself is now more than 35 years old. So it was one of the first, actually was the first database of that kind. And so we develop over all these years many various tools, and we collect them in geneXplain platform. So you click here, in geneXplain platform, and it opens for you these tools. And then you have here basically all the tools which are available. They are sorted in sort of categories, like depending on the data you have in your hands. And actually, it would be interesting to know what kind of data you have in your hands. So, for instance, if you're working with RNA-seq data, so if you're working with RNA-seq data, for instance, can you put the plus in the chat? So I just see if you're doing it. Oh, basically we have this pool. Thanks for organizers. So we have this pool. You see it now in your quizzes. If you click on quiz, right? So you can see this pool, and you can vote. So what kind of data, what kind of omics data are you analyzing? It's a multiple choice. You can select several of them. So either you have Affymetrix or RNA-seq or ChIP-seq in your hands, or maybe you have ATAC-seq, or maybe you're doing GWAS studies, so basically understanding SNPs and the association with particular diseases, or you're just doing genome sequencing of human or other model organisms, or you're doing proteomics, or maybe you're doing actually DNA methylation, so in your field in epigenetics, or maybe you are doing metabolomics. So all this kind of data, by the way, can be analyzed in geneXplain platform. And if you do this pool, so you can, we will maybe give you whatever 10 minutes to fill this, and when it's ready, so we will see the result. But in the meantime, let me actually. So I will actually cross. I cannot check it here myself, so it's only for you. So what I'm doing, I know, but I am interested to know what kind of data you have in hands, and depending on that, you might be interested in particular part of this geneXplain software. So I would but actually increase the font perhaps just to have it a bit bigger. So you see here, we can just work with all this kind of data I just mentioned, RNA-seq, microarrays, or you do the genomic analysis, you study genomes, you reveal mutations or variations in this genome, or you're doing epigenomics, DNA methylation, or you're analyzing microRNAs, so expression of microRNAs. All this kind of data can be loaded, and then you can analyze it, even if you just have a list of genes. So which means whatever study, maybe you just study literature actually, and you have your 100 genes, which very important for your for the disease you study. But then actually, let me put this chart a bit back. Yes, but then you can still use it here in the platform. Once you're here in the platform, you can load all this type of data. But what we are talking today is actually a very simple case. So you have your gene set, and you want to analyze transcription factor, predict transcription factor binding sites in this gene set. So the first thing, we want to load this gene set in the system. But before that, let me show you a little bit what we have in this system. Actually, we have different projects which you can create the projects by yourself, or you can use these projects, from already existing here on the system, in a sense, because you can look what kind of projects were already done before, and you can learn from examples here. In the example, for instance, you can learn what kind of analysis were done with a different type of data. Right. So you can open these projects and look at the data on that project, and read about, actually open any of those files, and also read about protocols, how these files were analyzed. But we will do it right now ourselves. So for that, let me go to one of the project which I have here. So for instance, this project which I created some time ago, and we can create here a folder. For instance, first TRANSFAC. And then, okay, so all right, so this project is limited for size. That's why it doesn't allow me to create here anything. So that's in those cases when you actually have a very limited project. But if you have your account and you get your license, then of course you will have a bigger space. So then I hope this project will allow me to open it. Let me check. All right, so otherwise we'll create a new project. Okay, my first TRANSFAC analysis. What I can create here. All right, so I have this folder now, and I can put my data here inside. So what kind of data do we have? And as I said, we'll analyze today just one example, which is a very simple data. So basically, we have this list of genes. Just opening it in Excel table. Let me open it for you and increase a little bit the font. So that's actually 59 genes, which are found to be upregulated by one simple experiment. So there's a TNF alpha, there is a HUVEC cell line, and TNF alpha was used to stimulate this cell line. And after such stimulation, after several hours of stimulation, the microarray experiment was done. That's why you see these Affymetrix IDs here. And basically, we did this in the platform, but today we are not showing how to find differentially expressed genes and reveal upregulated. These are all in the pipelines we have in the platform. But today we're starting just from a given gene list. So suppose you did this yourself in a separate, using separate tools or anything you like. And then you have this set. So you want to actually upload the set into the system and predict transcription factor binding sites for the promoters of these upregulated genes. All right. So then what we do here, we are staying on our folder, and here we have this import button. And we can import from different sources, even from FTP site or from just by typing your gene names. But if you have a gene set, you look at in the computer, and here we go to my example, and let me find this this file. So this is TNF stimulation upregulated genes. And it detects that in the first column, there is an ensemble ID. So, but actually, if you have your ID, which you would like to analyze and to be the first ID you are mapping them to genome, you can select the column you want to use. So for instance, you can use gene symbol as your ID. But of course, I mean, in case if you have such an exact ID like ensemble, it's better to use the ensemble IDs. All right, if you don't have any IDs in your table, or you're still not sure what ID to use, you can actually generate unique ID, and you get this table as it is, and then you can perform further filtering or further conversion of IDs inside the platform. But once you have the table with your IDs, you can directly import it here. All right, so we are here, and as we can see, here we have these 58 genes, and for instance, we can sort them by logfold change, and you can see this is the logfold change is very high, so like in 2 in power of 5, it's a quite significant increase of expression of that gene, and also p-values and also adjusted p-values are calculated, were calculated before, so you can see them here in the table. But so far, so we have just this list, and we want to further apply it to analyze your data. By the way, we have here also example, and you will see it in your account when you create your account. You will see it inside that we did like this first TRANSFAC analysis already before in January, and basically what we did here, whatever I was doing here, you see these results here as well. But okay, we will do it here by ourselves. So first, actually, we will go simply to this tab, which is called analysis. And analysis is presenting any tools or any pipelines which we have in the platform. And in the method section, we go now to the site analysis. And this here, you see quite a number of different tools to analyze sites. As I said, we have like more than 30 years of development. But for now, we need just this tool, which is called Site Search on GeneSet. And it has several parameters. What we're going to do, we just take this data and drag and drop it here. So let me open it again, drag and drop it here. And it's almost ready for analysis, but let us consider some of these parameters which we have to put here. All right, so first thing which we have to define is a so-called null set. And what is actually a null set? But maybe let's make a little interruption here to look a little bit into the theory of what we are doing here, right? So we're now returning back to our slides. Let me see where our slides are. And by the way, if you are ready with your pool answers, actually, we can see them, I guess. All right, so we'll see them. All right, what we see now, I guess you see it as well, right? Basically, 100% of you are working with RNA-seq. It's a very, very good result. And nobody is using Affymetrix arrays. That's right. So it's a pretty old technology, although it's very, very robust. Some using ChIP-seq, very good. ADAC-seq. Yeah, there is one somebody using GWAS, doing GWAS, sequencing, and even proteomics, and metabolomics and DNA methylation. All the panels. So you're actually in the right place. You can, as you can see, you can use all your data to analyze it, any of your data, and also in combinations. And we'll talk later, not maybe today, but in other coffee breaks, we'll talk also about how to combine different types of data. So if you have, for instance, RNA-seq and ChIP-seq, how to combine it in one analysis reasonably. All right, so very good. Thank you very much for your pool, for your answers. And now we are returning back to our slides. Let's just go a bit quicker. So this I don't need to repeat. You're doing your RNA-seq experiments. You know already what is going on with gene expression. Just to refresh your knowledge, we're always talking about TSS, transcription start site. So it's where the RNA is starting to synthesize, right? So, and this place in front of TSS is commonly called as a core promoter. But we also know that this core promoter is a place where most of the actually basic transcription factors are binding, but other transcription factors and also other like factors which are involved in regulation of genes, they are not only binding in promoter regions, they can bind in downstream more towards in introns or maybe in first non-coding exon. They can be also binding in very far regions which can be far from the gene, and they so-called enhancer regions, and they can interact with these factors which are binding to the promoter place, and then through this interaction they can regulate the gene expression. And always we are talking about transcription factors and binding sites where they bind. And these transcription factor binding sites, they are very variable. That's actually a very fundamental a principle of transcription factor regulation. So which maybe you underestimate, but that's really very basic and important principle, meaning that transcription factor binding sites are very short. And even though they are short, that they are also variable. So for instance, these are binding sites for a factor called AP1. And as you can see here, only these three nucleotides, TGA, which are standing in front, are more or less stable, but all the tail, it's very variable. So you can see many different nucleotides in those positions, which means if you would build any model of predicting these transcription factor binding sites, for instance, the model which we were talking today, these positional weight matrices, and allow to recognize those sites in genome, you will find many, many hits, because you have to allow not only the short region, but also you have to allow the variability. Otherwise, you will miss known sites, and all these which are shown here are known sites. A little bit, what are the transcription factors? So the transcription factor definition is very, very important and very flexible. Many people understand differently, but we have our own definition, which includes, in the first of all, the most important part of transcription factors is DNA binding. They have to have a DNA binding domain. They have to bind to DNA specifically. So this is layer one, we call it. This is sequence-specific transcription factors. But, we include also other proteins which are involved in regulation, but maybe they are not having this sequence-specific binding to DNA. So they're kind of adapters or the second level or even third level. So the second level we still consider as our transcription factors. So they're really often in very dense communication with a site-specific, with the DNA-specific part of the complex. But the other type of the cofactors, they may be already outside of this definition. So we actually include in transcription factors not only the site-specific, but also a few of them which are in very tight connection with them. All right. And the transcription factors, as I said, they have to have DNA binding domain, which binds to DNA. But they have also other domains which are involved in many different processes. And they are very, very important. For instance, activation domain, which is like a terra incognita. It's a very important domain, which in fact regulates gene expression. Because DNA binding domain is almost always is just to anchor the factor to a particular place in genome, but all the work is done by other part of the protein. But it's very little known about this type of domains. Still, since a lot is lot known about DNA binding domains, we have a very particular classification of those domains, of actually classification of all transcription factors according to the structure of those domains. And we have superclasses and classes, families, subfamilies, and you see it on example of, for instance, this transcription factor RAR, where you have actually various splice variants of this factor, which may be encoding from the same gene, but they're actually different proteins and they may play a different role. But if you classify them above, so on the upper level, you see families of factors which group several factors with a similar DNA binding domain, or which is then organized in classes like thin finger, like basic zipper, and several other classes. So you can see this full transcription factor classification on our website. All right, so this is an example of one factor which we mentioned, this AP1. And that's how we build matrices. And here, actually, as you can see, we present, so TRANSFAC is the first database which started to collect this kind of information. The positional weight matrices are there, but not only, as you know, TRANSFAC also collecting many other information about transcription factors, about known binding sites. But in the meantime, there's a number of public databases. Among them, you see Hokomoko, Jasper, FactorBook, Uniprop, and several others, which are also collecting the matrices by themselves. And TRANSFAC is, so we actually including everything into TRANSFAC. So after thorough verification of those matrices, and actually we built a kind of a reference resource for all positional weight matrices which are known so far. So then when you use TRANSFAC, you actually more or less not missing anything. So in terms of positional weight matrices. Now you have this matrix library, and then how to find transcription factor binding sites. We have algorithm internally, which I was showing before, so which is called MATCH. It was published many years ago. So all right, so I have a queue now. All right. So actually, this question is very important. I'm interrupting now because it's about what I was saying. Yes, the question is, does TRANSFAC DB include data on all the 3 layers about specific TFs? Means adapters and co-activators. Yes and no. It's a very good question, and maybe we will return to that. So there are a few second layer factors. For instance, one of them is very well known is P300, which doesn't have its own DNA binding domain, but it's cooperates with many transcription factors, and actually, and this set of factors it cooperates with have something in common. So basically, if you collect, we did collect all sites for those factors and build a matrix for P300. It's a kind of experiment of building a matrix for one factor, which is a kind of a cofactor. And it by itself, it doesn't have own DNA binding domains, it binds the DNA through other factors. But generally, we are not doing it for other cofactors. For instance, another factor called retinoblastoma, RB, which is cooperating with E2F and some other factors. It doesn't have DNA binding domain, and we don't have a matrix for that. All right. Okay. So coming back to the algorithms. So we have this much, and it quite specific algorithm. I will explain why. And there are other algorithms to find TFBS, and among them quite known is called Homer. And but not only, there are a few others, but actually the basic distinction is much in Homer is here. Okay, I will explain this difference a little bit later, but first let me go through a little bit how we build those matrices. Well, that's more or less a trivial. So we have those known binding sites, we align them, we count these nucleotides in each position, and this is called count matrix, right? Then we divide by, we normalize it to the number of sites observed, and this is a frequency matrix. And then once we have this frequency or count matrix, how we actually predict whether the binding sites is in that place or not, we scan it through the sequence, like this, and in each position of the sequence, we actually calculate a score. Well, in this case, you just sum up the observed weights in that matrix, like here you see T, 8, here you see A, 8, and you sum them up, right? So you have like a summing up the score. But it's not as trivial as this. So actually, there is a few theoretical backgrounds how to build a good scoring system. And the most most common scoring system is called log-odd score. And it's based on assumption that all positions in the binding sites are contributing to the binding of transcription factor independently. So they are independent from each other. So each kind of factor binds by each whatever amino acid or by each part of the protein to a particular nucleotide, and depending on which nucleotide is here, you have a little energy. It comes from this energy calculation, and then you sum up those energy contributions from each nucleotide, and you got this score. So, of course, there is much bigger theory. There are several papers written about how to come up to this formula. But in essence, you summing up kind of independent contribution from each nucleotide. That's one approach, and that approach is realized in many software tools. For instance, in Homer, it's having this kind of approach. But in the March, in TRANSFAC, we actually have a different approach, which is somewhat similar, but it has a little bit different score, which is important. Actually, we have this sum of the contribution of each nucleotide multiplied by a certain weight. And this weight is coming from information vector. And what is information vector? Is actually, it's also shown here in this picture. The bigger the letter, the higher is the information content of that letter. Basically, if the letter is small, you see this this nucleotide can be very variable. So if it's to end extent, you would have here 0.25, 0.25, 25, 25. So equal distribution of the nucleotides. In such a case, your information here is 0. If you have just like here, it's only one nucleotide is always here, you have actually information content equal 1. Well, actually equal 2. So it's 2 bits of information. And then we multiply the contribution from each nucleotide additionally by this information content, which gives additional weight to those positions which have a higher information. And that's in a sense taking into account not independency of the position in a sense, not fully, but still it's already towards including this not independency into the calculation of the score. So that's a whole difference. And as I said, in Homer, actually, they use this simple log odd score. So when which nucleotide is contributing equally. All right. Another important point which we'll showing today is selection of the cutoff or the threshold. And that's a very tricky thing. And even in this Homer user guide, they write that in fact the selection of this correct threshold, you may need guess and check. So although it's maybe not a very scientific approach, guess and check, but that's always what we are doing in sense. But in March algorithm, we found a certain process, a certain algorithm how to specify those cutoffs. What means cutoff? So when we, as I showed you, then when we predict the TFBS and we search for particular site in genome, then we calculate the score, and match return the score, which is from 0 to 1. So 1 means exact match to the best consensus of this of the site, which may even never happen, but so if the site is short, it might sometimes happens. And then the random match can have like a score around 0.5 or something like that. And then what we do, we actually apply this matrix, first of all, to the set of known sites. So for instance, we have 100 known sites, right? And then we apply this matrix and calculate score for each known site. And then we can put here the amount of, errors, which means amount of false negatives. So which means we, for instance, out of 100 sites, we miss 20, right? Because we put a particular high threshold, like here, which recognizes 80% of the sites, but 20% it doesn't recognize, because as I showed, even known site sometimes has very little commonality to the consensus, so they will be missed. So if you put very low threshold, which we call min FN, you will find all 100% of your sites. If you put a very high threshold, like we call it min FP, you will maybe find 1 or 2 of your known sites. We miss a lot. But in another site, we apply the same matrix to random sequences in genome. And we assume that by random chance, it will find most of cases false positive. So it will not find the true sites. And then at the same time, by increasing the cutoff, we, as you can see here, other way around. So by increasing cutoff, we increasing the false negative rate, but we decreasing the false positive rate. So we decreasing number of false positive sites. And by doing these two things, we specify the cutoff, which guarantees you to find most of the known sites, but may have a lot of false positives. Or otherwise you have a very high threshold, which means you will miss maybe a lot of known sites, but you will not have too many false positives, which means all your sites you found most probably are true sites. Then we specify this mean sum, which is somewhat sum of these two curves, and somewhere in the middle where you have minimum of both errors. All right. And then, we compared in one of our paper some time ago which approach would be better, basically using match or using this using approach like Homer. And then we call it additive or match-like score. And then we found out that it depends. So if you are searching for very very high scoring sites only, then the match approach is much better than the Homer approach. But if you search for sites with actually not losing anything, then maybe approach of additive is better than match. So that's you have to specify here. All right. Okay, before going further, let me, so I explain you the basics. So now we are coming back to our tool, which we now know what to do. So first, one thing I didn't explain, which is very important. Let me show it here. So why do you need no set? That's fundamental. Basically, everything is fundamental. Often what you are faced with, you have your set of 100 genes, your genes which are upregulated, and you want to analyze these genes, and you forget in which condition this happens. But you always need a control. So you always need something to compare with. And that if you do RNA-seq, and all of you actually are doing RNA-seq experiments, in your 30,000 or how many genes you have, you may find 100 genes which are upregulated at the top of your list. But in the middle of your list, there's a number of genes which didn't change expression. And imagine, these genes were there in the cell in the same conditions you were doing experiments, but they didn't change expression, which means they probably have or don't have something in their promoters which those guys who change expression have. So that's why it's always useful to compare. So we have this, we call it yes set, so set of your genes. And we always need a no set of genes we compare with. And here we have a formula. What it does actually, it does this important thing which is called enrichment. Why do we need this? So what is enrichment? So you actually see that these genes are upregulated, which means they have in their promoter something which is not here. Or maybe they have this something in more amounts. So as you can see here, like 3 of these genes have this green side, but none of these have it. So which means here you have like 4 together, but here 0. So you, and then you can calculate the p-value of such event. So if you would do it by, if you would observe it by random chance, of course you can observe it, but this probability of observing it by random chance is very low. So you can estimate using this formula. It's a very common formula, whether, you know, this observation of amount of sites you see in your set is higher than in the control set. So that's how we do it, how we estimate the p-value. All right. Now we are almost ready to do our analysis, but you see we need this null set. Where to get it? Unfortunately, I didn't have this non-change genes. Well, I have it, but I want to show you something else. You see in here in the public folder in the data sets, we have sets of genes which are especially for you who have forgotten to take these non-change genes. So as a set of the control, you can use actually housekeeping genes. You may now start, you know, blaming you are wrong. Those now housekeeping genes are also changing in many conditions, and you are always true. But still, you know, actually this set of 300 genes is published in one good paper where they scan many different conditions, and they really found this set of genes which almost always kind of stable. So that's a good set to compare. All right, so next, we need to specify your promoter length. And we have this from to, which means from TSS upstream, and to means downstream from TSS. So which means our default is 1,000 nucleotides upstream from TSS and 100 downstream from TSS. Such a region in which we, you know, from experience is most densely populated by transcription factor binding sites. And now the hard stuff. We have this profile. What is a profile? Profile is a collection of matrices which you want to search in your gene set with a defined cutoffs. And if you have if you look at the TRANSFAC database here, you can see here we have these profiles. And there you have a number of different profiles. Let me open, for instance, this one, which have 8,000 positional weight matrices. And this is called vertebrate human P001. What does it mean? So vertebrate means these are matrices which can be used to predict transcription factors for invertebrate genomes. So which means human, mouse, rat, all mammals, and maybe even down in the evolution, but all vertebrates we believe can be analyzed with these matrices. Of course, transcription factors vary in the between vertebrates, but they are quite stable. And sometimes in a close species, factor binding site, you know, DNA binding domain is even not changed at all. So, which means even the matrix was constructed on the human data, human factors. Still, to a certain extent, we can use it for any vertebrate genome. So this is the first thing, vertebrate. And second, human means, and human and P001, which means we select these cutoffs. There are two cutoffs. One is a core cutoff and another overall cutoff. And, you know, core cutoff is a cutoff for a very short region of 5 nucleotides in the center of a site. And the overall cutoff is actually the region of the cutoff for the whole site. All right. And we specify this cutoff in such a way that it gives approximately one hit for 1,000 nucleotides in the human genome, in human promoters, actually. So we have a human promoter composition of nucleotides, and then we can compute how many of these sites can be found. And then we put this score, this threshold, in this way that, you expect approximately one or a bit more sites in the 1,000 nucleotides. All right. So actually, to take this profile and put it here, you have to do it like this. And then there is one important feature, which is very specific for Match and also for TRANSFAC in general. Actually, I haven't seen it anywhere else, but it's very robust and very important, which is called optimized cutoff. So what does it mean? Actually, although we put these cutoffs, we still don't know how the transcription factor are binding to their targets in these real conditions. And as you know, transcription factor is binding to their target sites, not alone. It's not like a naked DNA and one factor. It's a DNA in a full of nucleosomes, and there is a lot of cofactors around. So depending on condition, the DNA binding property of the factor may change, right? And then it means we don't know actually cutoff for this particular case. We put a certain cutoff as a most, as a lowest approximation. But what is actual cutoff for this condition, we don't know. And then what algorithm is doing, it has these two sets, yes set and no set. And now it start changing the cutoffs from this minimum, which we put here, up to 1.0, and find such a cutoff, which gives the discrimination between these two sets, bigger discrimination, all right? So it optimizes the cutoffs on the fly for each positional weight matrix. All right, so we can put here, the p-value cutoff, and then that's it. So we can run this search. So in the meantime, basically, it's taking a little time. So you see how it works. By the way, you see here all the protocol, what happening. So certain promoters are extracted from genome, and it's the whole mystery how to extract promoters. So here we extract promoters based on TSS, which is reported in ensemble as a most upstream TSS of the gene. So actually a beginning of a first exon. And that's TSS, which is ensemble reports as most probable TSS. But of course, as you know, in different tissues, the TSS may vary, and that's, in some point, maybe 10, 15 years ago, that was a big discovery when people from RICAN were doing these, experiments of defining exact position of TSS for mRNAs. So they found out that depending on the tissue, the position is varying very, very, flexibly. Genes are different. For some genes, it's very stable. For some genes, you see a very big variability. And this tissue-specific TSS, we have this information in our system. It's stored in a database here, and we have a tool in our hands to analyze promoters when you know the tissue. And this tool is actually also available for you. It's for that you have to switch here at the upper right corner to a Match suite. So Match suite is a special tool, and we have we were talking about that before. Actually, it's also using quite easy user interface where you can analyze promoters in a tissue-specific conditions. So actually, you specify your tissue, like I'm interested in liver, and then it will pick up TSSs which are known for liver, plus it will consider the transcription factor expression, because also transcription factors are expressed differently in different tissues. and then it gives priority to those factors which are actually expressed in liver. And then, there are many other things to do, and this you will learn when you, you try this match suite, or you can read actually about this in our description of our tools. All right, so you see it's almost ready, this search. So it takes quite some time because I took the whole library of 8,000 position weight matrices, which, quite a big set. In fact, if you want to do it faster, you always can do the first scan by a smaller subset of matrices. We have actually two, historically, we have one set which is called non-redundant, with three different cutoff levels like mean FN, mean FP, and mean sum, which contains, if you look at this profile, which contains only 164 matrices, not 8,000, it's like 164. It will run very quickly, and you will see only the most important maybe factors. But actually, I would recommend you, if you have time, to run the whole set, and because for the same factor, sometimes one matrix works, in another condition, another matrix works. But so there is not such a thing like the best matrix in all conditions. Okay, so here we see the first results. So where they are quite encouraging actually, because what you see, out of 8,000 matrices, the algorithm found 1,700, so a lot, which have a p-value above 0.005 or 0.001, we put, right, and which have this significance. And moreover, you have here this yes to no ratio, which tells you the how much your promoters of your gene is enriched compared to the background. And by the way, this matrix, which is called CRL, is in fact here is 23 times more frequent in promoters of these TNF stimulated upregulated genes compared to the housekeeping promoters of housekeeping genes. So we can actually look at these sites by clicking here. So now I selected just sites for NF for this rel A matrix. And as you can see, actually, each line here is one gene, for instance, a promoter of one gene. So here you see promoter for CXCL2 gene, right? So this is a 1,000 nucleotide, and here the first exon, and 100 nucleotide downstream from TSS. And each arrow here is one binding site for this factor. And you see them here, many of them are near start of transcription. As I said, the closer to the start, the more significant is the match. And but if you can see, out of these 57 promoters, not all have those sites. So only this, how many? So have at least one site. Other don't have. But still, this is much more than we expect by random case. Well, there are other matches. For instance, if you sort by p-value like this, you see this, for instance, the first one, which is maybe very frequent, is but I guess this is a very short matrix, and we don't know much about this factor. It's is a zinc finger with number 392. It's very difficult to understand what it's doing. But second is a PO3, which is a known factor. It's of an oct family. And you can see that it's actually 3 times enriched. And if you now do it like this, you see a different picture. You see actually not all, but still many of these promoters have those sites. And some of them a lot of sites, like in this case, you see this promoter of this gene have one cluster of those sites in one position, which means it's those factors, they actually love this promoter very much, and they most probably binding to it very intensely. So this way you can actually see many of those factors. And actually, you can also get the a more colorful picture, of course, which you would like probably to have. So for that, let's filter us this table. Let's because it's quite a lot. So let's first filter it by p-value, which we would say that it should be below, let's say 0. Like this, for instance. And I would say we will filter it by also, I would like to filter it by the yes density, which means, if they are very frequent, so it doesn't bring you too much in terms of information, they just cover the whole gene. So then we will say, okay, it should be lower than say 2, right? So let's see how many of them we have. Okay, still 400, but we can still sort it by the yes to no ratio, and put this first 50 of them. Wait. Sometimes in Macintosh, it's not always working. All right. So we mark them all, and then we want to visualize them all together. So you see now these pictures of the sites, and interestingly, you see often these clusters, which happens, in some particular positions of promoters, which means it's not only one factor which is here. Well, you see it maybe in more details like this. But it also shows you that the same factor can be recognized by many different matrices, but not always in the same position. Some factors, some sites for the same factor are like clustered, like this case. You see NF-kappa-B matrices, and they all clustered. And here you see like Fra and June matrices, they bind here. P53 matrix bind in both orientation. So you see those clusters. But more about these clusters, we'll talk in next time when we will discuss the composition of transcription factors, which is a very important topic. Don't miss it next time. All right, so I would like to stop here. Just one thing I want to show you is that actually these sites can be shown here just as a in such a graphical interface, but they can be also mapped to the genome and can be seen in genome browser. We have actually here in the system installed a genome browser, so you can put the whole genome with all genes and all other information about the genome. But then you can, what we can do, we can take our sites. Yes, sites optimized. Let me actually open it again and drag and drop it here. And then you see them in the genome browser. You see this gene, and in front of it, we found a lot of binding sites. If you zoomed it more to the level of nucleotides, you see those sites found here. And as you can see, there are plenty of them found in the promoter of that gene. Many of them are overlapping. It's quite overwhelming. Of course, we have a lot of motifs, a lot of positional weight matrices. And if you are interested in one particular factor like NF-kappa-B, you can apply just for that matrix. But if you don't know which factor to study, you get such a picture. And then again, you have to come next time to listen how to find the most important combination of those factors using other algorithms we have here in TRANSFAC software. All right, so with that, I want to finish for presentation, and I'm open for the questions. One thing which I mentioned today is actually the Homer software. And you can, of course, find this Homer software anywhere else. But actually, we also put it inside our tool. So you don't need to search for it somewhere else. Actually, here we have a Galaxy section. And the Galaxy section is a section of tools which is connected to the Galaxy. Maybe you know this Galaxy package, and in our platform is fully compatible with Galaxy. So we actually including many tools from Galaxy package into our platform. And we also use this mechanism to include any new tool. For instance, we included this Homer tool, which is here. And you can use this Homer tool. It's available for you even without TRANSFAC subscription. If you just open your free account and this Homer tool is available here. For instance, here I did this. I analyzed this set of genes using Homer tool. And let me see where it is. I think this is a result of Homer. Actually, as you remember, we found like 1,000 motifs, more 1,700 using Match, and all 8,000 motifs from TRANSFAC. Homer is operating by a small library, and also it seems they put a cutoff much more stringent, and also they don't have this possibility to compare to the background set and to optimize the cutoffs. Anyway, so when you run Homer, you get like this result of 41 matches, so 41 sites for a few matrices. And one is a June B. It found these binding sites, and then stat, fra. Do we have also NF-kappa B? Unfortunately, Homer didn't find NF-kappa B, but that's kind of strange, but okay. But anyway, you can try Homer. Also, if you have any library of positional weight matrices, you can actually upload it to the system and use it here. There are a number of tools, and I had a previous webinar when I explained the free version of software of platform when you don't have TRANSFAC subscription. So what you can do, so you still can analyze your data, just for instance using Homer, or you upload some matrices which you have, you get them from internet, from some other libraries, and then you can clearly using them here with all advantages, also using this optimizations of cutoffs and everything which is available here. You can use also with any foreign matrix collection. All right, now I see some questions. All right, so first question, whatever analysis is done, is there a summary report generated? All right, a good question. Actually, today I showed to you how to do analysis just using these tools. We generate various types of outputs, like tables, and first what you see, you should know that anything you get here is exportable. So you can export it as a top separated table, or actually you can export results as a match report. So for instance, where we had this, for instance, when you see this, you can actually export it. Let me see, export. Right, so you can export it as a BAD file, so with all positions of the sites, or as GFF or VCF. If you have sites, you can actually export it as I think that we have also standard format for the match export. So you can export everything by itself. Some of the tools and especially pipelines have a small report. So for instance, here, if you run, I run it as a separate tool, but if you go for the pipeline, which does a lot of things for itself. So for instance, here we analyze regulatory regions, identify and enrich TFBS using TRANSFAC. And then there is a pipeline which does a lot of things. So if you have your gene sets in the wrong format, it will convert it in the correct format. It will also extract transcription factors and do many other things. And it actually generates, some of those tools generates a little HTML report. I can show it maybe, if I find it now. But also other tools like MatchSuite, for instance, it's generating very good report. So like PDF of that report can be downloaded. It's colorful. It has a lot of pictures, tables, and everything. So try it when you have time. And if you have account here. All right. So that's about report. Okay, second question. Will there be any session on microRNA target prediction in future? That's a very good question. You actually give me a very good idea. Yes, I think we should do it. I guess we now plan all our seminars maybe until the beginning of summer, but somewhere in summer we can make this. Okay, let me write it down, microRNA. miRNAs, that's very important actually. You have a couple of tools which are very specific in our package. I will not take now time for but just to show a little bit, we have this microRNA collection of methods, and some of them like this microRNA with feed-forward loops is very specific algorithm, which is it's very robust and very interesting. Anything, and very important actually, one thing you should also know that microRNA, it's you downstream from them, it's not much known. So of course you may know target genes, but that's all. So many microRNAs are still not clear what they are doing. So you cannot, for instance, put them in geo categories or into disease categories very rarely. But they all have promoters, right? So they express from some genes, and then those promoters are available for analysis of transcription factor binding sites. So in that case, in that respect, our tools are really doing very unique type of analysis of microRNAs when we analyze promoters of them and understand how these microRNAs are actually regulated, why they are upregulated, for instance. Okay. All right, so I think we don't have more questions for today. And with that, I would like to thank you for that you came here to listen my talk. And I hope you will come for the next coffee breaks. And don't forget to put your questions. I will be very happy to answer them. Thank you very much and see you next time.